pull up a chair, take out the oxygen, and join the discussion.
You've just stepped into Life Unculturable,
the world where conversations grow faster than colonies.
We're your hosts, Sam and Julia, scientists by day,
Podcasters by night,
and professional oxygen avoiders 24/7. Join us in exploring the science of microbes that thrive in the absence of oxygen on Life Unculturable.
Well, Sam, it's good to be back here with you.
It is good to be back indeed. They let us back in the building, so that's always a plus.
They did. Maybe they don't know what we're doing in here.
That's true. If somebody else stumbles upon these audio recordings, we'll be in trouble. We got to make sure to delete this when it's all said and done.
Delete all evidence.
Delete all evidence. I like that.
Yeah. Well, How are you doing today?
You know, I am doing pretty good, all things considered. How are you doing, Julia?
Pretty good as well, all things considered. I'm well fed, well caffeinated, and ready to go.
All right. Well, I agree with that. So I think on that note, we'll just jump right in as we're talking about aerobes versus anaerobes, a tale as old as time, the battle between Beauty and the Beast, you know, the armies dueling one another. I've got Mordor and Frodo and Gandalf, and we're gonna get in the nitty gritty of things today, folks.
Are bacteria Frodo or Mordor? What is worse?
They're probably like the orcs,
like sprouting out everywhere.
Yeah, something like you can't get rid of them.
Oh yeah,
they're everywhere, and that's probably good. It's probably a good analogy.
Nice.
But yeah, before we dive in today and start just hammering people over the head with all things anaerobic, how would you describe what it means to be a strict anaerobe? You know, to those people out there that maybe don't deal in strict anaerobes that often.
I would say a strict anaerobe is an organism who cannot live with the presence of O2.
Ooh, so molecular oxygen. We're talking that good old atmospheric.
Yes. That always tripped me up when people said like no oxygen, and I'm like, but the molecule oxygen is still there in anaerobic environments, and anaerobes can live there, but the O2 is the enemy.
Right, just the O2 by itself.
The O2 by itself. Yeah. And strict means it really does not like to be exposed to it at all. But I also know that there are different kinds of anaerobes. Like there's different levels to it. So some can be exposed short-term, have like some adaptations that they can deal with the short-term oxygen exposure. Some can even deal with more longer-term oxygen exposure. And then there's the in-betweens, the ones that couldn't decide which side they want to belong to, the facultative ones.
Yeah, no, that's a good point. There are several different types, and even some anaerobes that just give up and they're going, okay, I'm going to produce an endospore now and just wait for better times.
Yeah.
And so there are a bunch of different strategies out there that they tend to use.
Yeah, absolutely.
Yeah. And speaking of strategies in anaerobes, are there any particular precautions that we would want to take to cultivate these strictly anaerobic organisms in the lab?
I mean, the major precaution would be like to get rid of as much atmospheric oxygen as we can. And there are different strategies we can do that with. Some require equipment, some don't really require any other lab equipment than any aerobic lab would have. Yeah. For example, boiling your media can get rid of the dissolved oxygen, and any lab can put a flask onto a heating plate and boil it, right?
Totally. Yeah, I think it's all about getting the oxygen out and then effectively keeping it out once you've gotten rid of it. Yeah, sometimes that's almost more the struggle.
Probably.
I'm imagining like tubes with rizazirin somehow turning pink afterwards, and no, not again.
And maybe for those who don't know, rizazirin is one of the color indicator we use for the redox state of a media.
Yeah, that is something you do not want to see pinkify, people. That is.
Yeah, or red.
Yeah, that's a sad day in the life of an anaerobic microbiologist.
Absolutely.
Yeah, best to just not use it and hope for the best. You don't have to worry about seeing it then.
If you don't know, you don't know.
Exactly. Although I will say in that vein, Resazrin, in my experience, is only really beneficial in lightly colored media.
Yes.
So if you've got, kind of like a watered down Mountain Dew or, it's a lighter colored media, like it's probably fine, but you start getting into something that's a bit darker. And then at that point, it's like, what is brown plus pink? It's just still brown to me.
Yeah.
Like there may be a slight darkening of the media, but it's very, at least for me, to tell in darker media than it is for something that's lighter in color.
Yeah, absolutely.
In general.
And there is a point where any number of like holding it against the light would not help you. Like pure rumen fluid, for example.
Exactly. Good luck with that. Yikes.
Okay, so we had like boiling of the water might be something we do that aerobic folks don't do. We also flush the media, is what we call it, making sure that the headspace does not have any oxygen by instead blowing nitrogen or carbon dioxide or any other anoxic gas on there.
Yeah, totally so. So that being like that headspace area, essentially just the portion of your tube. In our case, we're typically dealing with like serum sealed and crimped tubes. Yes. And you just want essentially the part that's not liquidy to also be oxygen devoid. And so, we try our best to get the atmosphere in the tube devoid of oxygen and then the actual medium itself devoid of oxygen.
Yes.
Yeah, and I think once we're in that state, we're golden.
We are.
For the most part.
For the most part.
Yeah, that's when the difficult stuff begins. Then we got to grow things. What
kind of materials are like gas proof? Like our tubes, for example, are made of glass, right? Thick glass. Rubber might also be rather gas proof.
Yeah.
But if we're getting into the plastics.
Yeah, plastics are tough. And even working with plastics in, say, like the chamber, for instance, you're going to be packing in essentially like oxygen's clung onto the plastic as and when you take, say, consumables into the chamber. And so for me, I like to, if I take in like a bunch of serological pipettes or, sterile loops or whatever the case might be, I like to give them a good just waiting period in the chamber so they can interact and work with the catalyst and do all of those things that they're going to do for a solid 12, 24 hours before I really go in and do anything of importance. Yeah.
Interesting.
Yeah.
Oh, and maybe for those people who have missed our first episode, chamber, what kind of chamber are we talking about here?
Well, I'm talking about an anaerobic chamber. The way that I would paint this picture for you, if I was Claude Monet himself, is I would tell you that it is a gigantic, oversized vinyl balloon with armholes. And essentially that vinyl balloon is puffed up with anoxic gas. And so for us, or I should say for me, because I think we run slightly different blends of gases in our chambers, because you fungal folks are all about CO2 and I'm more or less nitrogen and hydrogen. But essentially what we do is we pump those chambers full of something that doesn't have O2, and then we let it set, and we let these little catalyst boxes that we have in there turn any residual oxygen into water vapor, and we scrub that out with things, and then we can get the party started with the anaerobes.
Yeah, nice.
Yeah, and so it's handy to have, but it's also not always necessary. Like you can certainly get by, working with very strict anaerobes using things like, say, a gassing station, for instance, and some good Hungate technique. It tees back to one of those, papers that came out long ago that still holds up today, that we're still utilizing and still very much valuable in our field.
Absolutely.
Yeah.
And then the chamber, going in and out of the chamber is also quite a process because you, of course, cannot just open a hatch and not care, right? It's like you fill that balloon up with anoxic gas and then you just open it up again and don't care. That doesn't work. So you have this little airlock. And people who have listened to our intro maybe can hear the sound of that airlock.
Yes.
In the introduction.
Yeah, that was not us mimicking noises. That was a microphone, I believe, inside the airlock that we put through a good cycle.
Yeah.
And essentially, what it's doing in that moment is it's taking whatever atmosphere is in that enclosed space in the airlock and essentially vacuuming it out and then pushing in, and again, an anoxic gas, and it will do that several times. And in doing so, what we would call that is like a headspace exchange. And so it's, again, just sucking stuff out, spitting in nitrogen. Sucking stuff out, spitting in nitrogen. And then in my case, it ends with a nitrogen-hydrogen blend. And so I get a little bit of a mix there at the very end before I open the door on the other side.
Nice, yeah.
Yeah.
Well, we're off to a good start.
We are.
So.
We've been here a solid 5 minutes, and we haven't spent 50 minutes trying to figure out the audio tracks.
True. Which we, of course, haven't done the last 2 episodes. We're professionals.
We are professionals.
We are.
And so something that Julia and I decided might be a fun thing to do, let me whip open this laptop real quick, is to try to do a blind ranking segment here today, folks. So in this blind ranking segment, essentially what I'm going to do is Julia has absolutely no idea what these things are. I'm going to give her a topic here in a minute, and then she is going to attempt to rank some things for us. Now, the rules are that once you rank them, this is going to be 1 through 5, no takebacks. So it's got to stay where it is. And so we're going to start off with microscopy techniques.
Oh, wow.
So what I'm going to do is I'm going to give Julia some microscopy techniques, and she is going to rank them as number 1 being the most important to her and her work, and 5 would be the least important to her and her work.
Importance, not enjoyment. Okay.
Right. Importance, not enjoyment.
Okay, got it.
So here we go. Without further ado, we're going to start off with SEM.
Ooh. Damn, that's a hard one. Importance, not enjoyment. Because I enjoy SEM like a lot. But importance-wise, maybe what other things do you have on that list?
To be determined.
Let's give it a 4 in case there will be something even less important to us. Although, I mean, have you seen those SEM pictures of fungi? They're amazing.
They are absolutely wild.
Yeah.
But you enjoy the SEM process, all the ethanol washes, and that it takes 5 days.
Okay, I should clarify. I enjoy sitting at the microscope and taking the pictures. And I enjoy looking at the pictures.
Okay.
Everything before then, we have some students who do that.
You did? I will say that I agree with you. I love sort of being the white knight that swoops in at the very end when they're like, oh, Sam, we're getting ready to look at it. And I go, yes. And then I just forget the fact that they've spent 8 hours prepping these things.
Yes.
Okay, so we got SEM coming at number 4. Next up is Brightfield.
Well, for fungi, that's pretty much up there. So I would give it a 2 because you will already be able to see, like if you're dealing with a bulbous or a filamentous fungi. And for anybody who does not know what that means, stay tuned because we will have an episode explaining all of this, and I will never shut up about it. So, yeah.
Okay,
number
so
2.
we got that one at number 2. Then I'm going to come in with fluorescence.
General fluorescence or like stains, fluorescent stains?
I was going with like fluorescence microscopy. In my head, I had you with like when you do your nuclei staining and
oh yeah, okay, so
things of that sort.
That is also where my mind immediately went, although I spent half my PhD doing fish for anaerobic fungi. But no, number 1, definitely. We need to know this nuclear localization.
Okay. And then the next one I have is confocal.
Well, I mean, there's not much information to be gained because our fungi are pretty big and usually well contrasted in bright field. So, meh. What numbers do we not have? 3. I'll give it a 3.
All right, 3. And so that leaves by default in the number 5 position, the face only a mother could love, phase contrast.
Okay, yeah, that goes right at number 5 because it's neither important nor enjoyable.
That's so funny. That's sort of why I wanted to do this one last. I was like, I don't think that's going to be very important for Julia. So we've got fluorescence coming in at number 1, bright field at number 2, confocal number 3, SEM number 4, and then phase contrast number 5. I think for me, if I was going to have done this, I probably would put fluorescence at 5 because I don't really do a lot of fluorescent microscopy, confocal in the 4, and then at the 3, phase contrast, bright field number 2, and then SEM number 1.
Oh, interesting.
Yeah.
So it's like you want the surface structure of the bacteria?
Well, not necessarily even the surface structure, but for us, I think size and the actual cell,
Oh, because they're
like
so
the size
small.
and the shape, and whether or not it produces an endospore, and if it does, is it a subterminal? Is it where is the endospore located? And I think for us, the royal us here, for me, I'll say for me, and so I think for me that's a really important piece of technology that gives us a lot of really good, really solid information. Whether it's the grouping of the cells, I can't even think of the word that I'm trying to say here. Like we got pairs or tetrads. There we go. And then, again, size and that sort of thing. Flagella, getting to see those flagella on the SEM too, and where they're located, the relative lengths of them. Like I think it's a pretty powerful tool for us.
Oh, and so,
humble bacteriologists.
and the standard microscopy, so to say, like the phase contrast or bright field microscope we have, that's just the magnification is not great enough for you guys to really distinguish those facts?
Yeah, it's not necessarily the best. It's good for some things like, gram staining. Is it, gram negative cell wall type, or is it a gram positive cell wall type, and things of that sort. Or even getting at, like, is it modal? Like, can we watch that sucker swim through the media and that sort of thing? Or just checking for, like basic purity, contamination, things like that in cultures. So it's nice for some of those reasons, but for getting at the, the nitty gritty structures of things, I'm SEM all day, especially if I don't have to prep the slides. So I should preface with that. I might knock SEM off the board if I got to prep these things. But,
well, interesting. Hey, but that might be the first thing that our fungi do not suck at. Because usually when I listen to you, it's like, oh, I toss them in the fridge for a couple of months and then I grow them again. And like, I'm so envious most of the time. But hey, our fungi are big and they're easy to spot. And you don't have to prep SEM samples to really distinguish morphology there.
Yeah, well, I mean, I really do. I love the pictures of all the microscopy pictures that you share with me, whether it's on ours upstairs or whether it's the SEMs that we go and do downstairs. I don't think I've seen one yet where I go, oh, that's not super cool. Like I love it. And when you guys put those figures together that are, it seems like there might be, 15 or so, panels in a figure.
Figure one.
Yeah, figure one. Dun, dun, dun. I just love those because there's so many different and just wild looking structures, whether it's the zoospores or the hyphae or just whatever it is. It's always so crazy looking to me. And so for like the bacteria, it's like, is it a circle or is it a pill shape? Like every once in a while you're like, oh, it looks like a corkscrew, but that's all we got really in a nutshell. But with the stuff that you show me that you guys are isolating and pulling out into culture, it's always so wild. And maybe to you all it looks like, oh, it's just another Pyromyces or it's another anaeromyces or whatever it is. But I look at it and I go, man, that's wild. Like it just blows my mind. I'm always impressed by the microscopy images.
Nice. No,
Yeah.
I'm the same. I'm still sitting at the microscope and just not wanting to let go and go home if I look at them.
Yeah. Oh, I know. I can't think of the amount of times that someone's come into the lab and been like, oh, Sam, have you seen Julia? And I'm like, she's probably at the scope. I haven't seen her in like 3 hours. So, you might check the microscopy.
Yeah. It's
The
my
microscopy
hiding
room.
spot.
Yeah. Oh, well, in that case, I'll be like, she's gone for lunch. Come back around 4:45.
Tomorrow.
Yeah. No. That was good.
Nice.
I think I respect those rankings.
Yeah, thank you for putting that together. I enjoyed ranking it.
Well, fret not, fair maiden, for I have yet another.
Oh, you have more.
I have 2 more of these.
Okay, let's
partially
do
in
that.
case the first one didn't work, but I suppose we can always cut these out and put them in some other place. Now,
I enjoy talking to you a lot and it's easy, but let's, for our listeners, maybe step back and think about oxygen is all around us. So where the hell do those anaerobic organisms live if they cannot tolerate oxygen?
well, I think you can find, and we alluded to this in the first episode, that I think you could throw a rock almost anywhere and then find an anaerobe close to that rock. So I think typically when I'm thinking about anaerobes, I think about sort of 3 categories. I think about host associated, I think about environmental, and then I think about sort of engineered type environments. And so I think especially host-associated are the ones that tend to come to mind, like the human gut, gastrointestinal tracts of fill-in-the-blank animals, even things like the oral microbiome, like we find anaerobes on our teeth, a lot of like fusobacterium and prevotella, and some real strict anaerobes like in your teeth, and or on your teeth, I should say. And so I think you find those in host-associated little niches, and then environmental, definitely soils, aquatic samples, water, that sort of stuff, and even in things like engineered environments, like I said, they're all over the place.
So essentially anywhere. Is it only bacteria though? Like, are there any other life forms that we know is also are also anaerobic?
Well, we know thanks to you that there's obviously AGF.
Yeah,
so we've
hint,
got so we've definitely got anaerobic gut fungi out there. But yeah, definitely there's what comes to mind for me are things like archaea. So I think about a lot of methanogenic archaea. The sort of the poster child of that's probably like a Methanobrevibacter, one of the more common archaeans in the human gut. Essentially one of those methanogens that's really producing methane by essentially metabolizing hydrogen and carbon dioxide. There's certainly other even domains of life that are out there that are, anaerobic in that sense. And even some other, eukaryotes as well, like protists and the like. So, yeah.
Yeah. So we know where they live. We know they don't like oxygen, but what is the problem with oxygen? We're fine.
Allegedly.
Mostly.
Aren't we really fine? Another philosophical debate there. Yeah, I think for the most part it boils down to oxygen toxicity, things like reactive oxygen species, some ROS. These ROS are essentially, think of them as like hyperactive sort of byproducts that can damage things like proteins and DNA, and essentially certain organisms have the capability to deal with these reactive oxygen species, and whereas others don't. And so they might possess particular enzymes like superoxide dismutase or something, very technical like that allows them to deal with that oxidative stress. So that way they don't essentially succumb and just die when they come into contact with it.
Yeah, absolutely. And people outside the science field maybe heard about ROS and oxidative stress in the context of aging. So it is like ROS and oxygen are not only toxic to anaerobes, they're also toxic to like aerobic organisms, but aerobes have just evolved to deal with it.
Totally. So that's what's Never mind.
Oh,
that was gonna be bad.
I want to hear that at least.
I was like, so is that what's gonna be giving me wrinkles? Can I blame Ross for my wrinkles?
Oh, we can always
curse
blame
you,
Ross.
Ross.
Yes.
We were on a break. Oh no! Rein it back in, folks. Come on now. One of the recurring segments that Julia and I keep going back to is the questions from the audience, and again, for our audience, we just keep going back to the undergrads. And this is the Q&A from the undergrads. And there were actually a couple of really good questions this week, so bonus points to them for conjuring these up out of the abyss. And one of them was, how much of anaerobic work is science versus superstition? Are we superstitious scientists?
Oh my God. I guess as humans, we cannot be otherwise. And I am super superstitious when it comes to my lab work. Like there are some things that just need to happen a certain way, so I can convince myself that will then work.
No, I couldn't agree more. And even in the ways in which we do things just a smidge differently. Like for instance, if I'm making my bulge tubes and I'm doing sort of like the Hungate technique and getting things going on the gassing station and outside of an anaerobic chamber. Like I'm a big proponent of like vacuum grease on the the rubber stoppers to go and seat down in the bulge tubes. Mm-hmm. And one of my superstitions is that after I've crimp sealed everything and it's all good to go, I will then take my finger that has the the vacuum grease on it, and I would like double tap the tops of all of the tubes. It's just like, good boy, good girl. Like I just go through and I double tap every single one of them, and that's totally backed by science. We have really great p-values.
Yes.
A tube's never exploded in the autoclave.
Yes.
That I know of after having done that. So yeah, definitely there's a bit of superstition that I think
absolutely
a semi-healthy amount.
That's similar to when I do the culture collection transfer. I am convinced that our fungi need a certain kind of a mix between love and neglect. They need to feel that you love them, but you also hate them. And this right in between when you do the culture collection will ensure that they grow. I'm convinced of it. And I know that some of my colleagues are too. Because our fungi, they sometimes, and I think that's where most of the superstition comes from. There's so many little factors in all of this lab work, especially in anaerobic work, but also if you go down to the molecular level, like PCRs and transformations and whatnot, that if there's a little bit that goes wrong, there's so many factors. If you have a little one of those factors going wrong, your experiment fails. But you, of course, cannot keep track of all the factors at once. So sometimes you just don't know what's happening. And then your brain needs an explanation. And my explanation is definitely that our fungi that like to die on us sometimes, if you love them too much and if you neglect them too much, they're not good. But if you hate them the right amount, they will be fine.
Just the right amount of hate, folks.
Yes.
I think that's pretty spot on. I don't know that I approach most of my anaerobic bacteria with hate. It's more cautious optimism. Oh, come on, fellas. Let's do it right this week. Because I've recently went through a spell in the lab where it seemed like absolutely nothing was growing. I talked with you about that the other day off air, and essentially all of the media that I have made for the last, call it, 10 days, essentially nothing worked. E. coli wasn't growing on TSA, Enterococcus wasn't growing on bile esculinase agar. Absolutely nothing was going on, and so I blame witchcraft and wizardry.
Oh yeah, absolutely.
That's all that I've got. We had a full moon the other night. I think things are back to normal now. I'm at least seeing dots on plates, so I'm happy with that.
Jupiter was in retrograde.
Yeah, I mean, I'm sure it was probably something a bit more technical than that, but I'm going to chalk it up to superstition and alchemy.
Absolutely.
Somebody casted a curse on my bacteria.
I also once worked with a colleague who was convinced that if she doesn't do her transformation, like we were trying to transform yeast cells, if she doesn't do those transformations with the specific set of pipettes, they won't work. Which just goes to show that this is a universal truth. It's happened more in anaerobic work because there's even more factors that you can control. But it's definitely true for molecular biologists as well.
I think it's interesting that you bring the pipettes and the molecular side into this, because I totally have a superstition about which multi-channel pipette I use when I'm setting up a PCR. And because we have 2 in the lab, and one, I think it's got 8 channels, and then the other one has 12. And the one that has 8 channels for pipetting small, sub 5 microliter volumes, I absolutely detest the 8 channel one. I would not be caught dead using that one. And for no other reason than at first I thought, the 12 channel one, it's prettier. Oh, I like that one. And then over time is kind of like you alluded to earlier, like a PCR fails or, something happens with the gel and it smears, or, it's an ugly gel. And I'm thoroughly convinced now with a healthy amount of superstition that that's the only multi-channel for me. Is that blue 12 channel multi-channel pipette.
Oh yeah.
Yeah.
I love that one, by the way.
I know.
It's that so one nice. Is just and 12 is just, that's the right number of channels to have. Right. What is 8?
I know. You get 4 more. Yeah. Yeah. No, that's good. That's good. And so shifting gears, the other question that the undergrads sort of threw our way, sort of a more, we'll call this one opinion-based, And it's if aerobes are the golden retrievers of the microbiological world, what kind of a pet are anaerobes?
So the first question was not opinion based. Like that was pure hard facts.
Yes. Yeah, I think the consensus is we're superstitious, so
Yeah.
I see no reason that that's opinion.
Okay, I can get
opinionated.
behind that. Golden retrievers.
Because I guess we're assuming the golden retrievers are nice. Perhaps you're not allergic to dogs. You know, we'll preface by saying a golden retriever here is the epitome of a fantastic pet.
Oh, okay. Good question. Because I would have thought that aerobes are more like, I don't know, fish or triops. Like in German we call them Urzeitkrebse, which means essentially prehistoric crabs. Because you just dump them into a nutrient broth and they just flourish and you don't have to do anything else. But, what pet are anaerobes? Probably one of the more rare reptiles you can have. I mean, I've never owned a reptile pet, but I've seen the Instagram videos of people who do, and then the temperature has to be just right, the nutrients have to be just right, they need to be fed a certain thing, or I don't know, and there goes so much care goes into those pets, those more rare reptiles that I think maybe that sounds a bit like our annoying anaerobes.
I was going to say, especially the AGF, because they're, from what I've learned over the past year being around you AGF folks, is that temperature is almost everything.
Yes.
There is not a lot of deviation around what's optimal versus what is the like the actual range for these things to grow in. It's just a couple of degrees different.
Yeah.
Everything's dead.
Yes.
And I know for me, it's like, if it's optimal is 37, I can do 40. I could probably get away with 30. I could do 45. I probably won't go above 45 or below 30, but like I've got a good 15 degrees Celsius that I could try to grow these things in. And I think with you guys in the AGF, it's, you've got maybe 5, maybe. It's usually a pretty tight window that you have to keep that collection at.
Yes, that's true.
Yeah, man. So if aerobes are golden retrievers, boo. I like the analogy of the exotic reptile, but I think I'm going to call anaerobes sort of like one of those kind of like hairless cats. Because I think that they're sort of like a face that only a mother could love, and and the people that own them are proud of them, but I'm not into them. And I think oftentimes with anaerobes, it's like the people that work with them are really prideful. They love their C. diff. They love their, whatever the Methanobrevibacter, whatever strain it is that they're working with. It is like their hairless cat that they're so proud to show off, to talk to everybody about, to give them life updates on. Like, it had its first hairball. It's such and such years old now, and everybody else is like, ew, that's ugly. Just get it away from me. What are you talking about? And so I think I'm going to give them that moniker. I'm going to say that they're sort of those hairless cats. But to be fair, I don't know that I really like those hairless cats, but I do love my anaerobes. So I suppose I fall into the hairless cat
Absolutely. And
portion of this.
We will complain about anaerobes all day long, but we will fight and defend them because they're just the best. And with the best, I mean the anaerobic fungi, of course. But I think, your hairless cat is a prokaryote.
Yeah. No. And I will say that's something that I try to do, like at the MCCF. So for those of you that maybe didn't catch that part of the first episode, the MCCF is the Microbiomics and Culturomics core facility here at Oklahoma State University, where I spend a good chunk of my time. As we're consulting with clients that have particular desires and wants with respect to like growing bacteria from particular samples, oftentimes those individuals are almost entirely focused on the aerobic fraction of whatever it is. And so I'm always advocating for, hey, how about those anaerobes? We should try to grow some of those. And almost to the annoying side of the spectrum sometimes, but I try not to be overly annoying.
Oh no!
But I
sure
definitely
you never
do.
are.
No, me? No. I gently nudge until I'm told not to.
Nice.
Yeah. Well, those were a couple of really good questions. We might have to throw the undergrads a bone, maybe some leftover Halloween candy or something like that.
Sounds good.
Yeah.
So aerobic organisms are using oxygen in many of their cellular processes, and mainly glucose is broken down essentially with oxygen to ATP, which is energy for the cell, and anaerobes cannot do that. So what do anaerobes do instead, and how effective is it?
Well, I would probably start with the end of that question, and I would tell you that my bias is that they're a smidgen less effective at utilizing glucose, in a sense that if we're just talking about pure output of ATP, I stick one glucose in, I get 2 out with an anaerobe, 2 ATP out. If I stick one glucose in with an aerobe, I get 36, 38 ATP out. Mm-hmm. And so just purely looking at it from that perspective, it's like, which one do I want to invest in? I would want to, I think, invest in the one where the return is so much higher per molecule of glucose, just in general. But yes, essentially what they're doing, if we're talking aerobic versus, anaerobic, that if it's aerobic, they're using oxygen as that final electron acceptor. Yeah, I always used to tell an analogy when I would teach this in some of the labs that, sort of imagine me in 10th grade in high school, like falling down the stairs. The further I fall down the stairs, the more mad and embarrassed I become. And if you think of these metaphorical stairs as the electron transport chain, as you're bouncing down it, and ATP is just popping off all over the place. That's sort of the picture that I set for when that happens.
That's quite the picture.
Yeah. I'm based on a true story, folks. Not that they're going to receive detention for that or anything of the sort, but that's a story for another day. Anaerobes, they're not going to be using that O2 as a final electron acceptor. They're going to be using things like sulfate and nitrate, pyruvate. They're going to be using something that's not simply just O2. Mm-hmm. And I think there tends to be sort of a feel, the royal we here, we microbiologists tend to look at these as sometimes being less efficient for that reason, just because the yield is so disproportionate to us. The 2 ATP versus, say, close to 38 ATP per molecule of glucose. And, yeah.
Okay, so the argument would be that there's less biomass, less energy available to produce biomass or
perhaps,
whatever,
and even
or
slower
proliferate.
Growing organisms sometimes.
Mm-hmm. Yeah.
Although there are some fast growing anaerobes.
There are, yeah. And I don't know, my mind, my critical mind heard that story of because of course I learned it during my studies as well. And that story about the ATP produced is really neat and nice, right? Every evidence that we had since the 1930s kind of put together that story of, hey, electron acceptor oxygen is just the optimal one to use. And that's why it evolved over time. But I feel like maybe we just don't know, we're biased towards that story. We like that story. It fits so nicely. But we cannot deny that there's fast-growing anaerobes, and there's even slow-growing aerobes. And these kind of two pieces in my mind fit together better if the argument is not anaerobes have less energy because they produce less energy, and then they cannot grow as fast, but rather maybe the nutrient density in the environment will always determine the growth rate more so than the presence or absence of oxygen, because anaerobes have evolved for, way longer than aerobes have, and they seemingly are fine, and there's a huge diversity, probably way bigger than we currently know, with huge adaptations that we are currently bit by bit discovering. And they might not use glucose in the same way that aerobes do, where you break it down, but they have other adaptations that they use. They can like some of them ditch, for example, the production of proteins and everything from scratch, and rather have more importers that would take up proteins and parts, and so they don't have to have enough that much energy because they're not starting the process from scratch, like aerobes sometimes do.
So are we comparing aerobes and anaerobes to like first year versus final year grad students? I am so sorry if that is you that is out there. That is the only thing that popped into my brain in that moment. Is starting stuff from scratch and efficiency.
Yeah, probably. So an anaerobe is probably a postdoc or a PI who has like other people working for them.
Hey, there we go. I like the way that's set up. That's set up nicely. Yeah.
Yeah.
Yeah, it is very costly though to synthesize, certain those proteins and all those other cellular components. So,
Yeah.
Yeah. Yeah. And I think even in that sense there's a lot of, just due to that sort of breadth of diversity that you're talking about, it makes anaerobes sort of attractive in a sort of a biotechnological sense, because there are certainly a lot of processes that anaerobes are pretty good at, that we're attempting right now to harness to make things better, or to be more efficient in the production of fill in the blank, whether it's bioethanol or acetate or whatever other anthropogenic compounds that we find useful to us, where we need something to happen in sort of an anoxic setting like that.
Yeah, certainly.
Now, the last ranking segment here, essentially I've got 5 random things. These are all sort of categories of microorganisms. Think of them as a fill-in-the-blank file. They love a particular type of environment, and Julia is going to rank 1 through 5, coolest to the not coolest extremophiles.
Ooh.
Yes. And so 1 would be what she perceives to be the absolute most cool extremophile on the planet. And 5, we could just get rid of it. Cross it off of the endangered species list because it ain't with us anymore. And so we'll start off with acetophiles. So some acid-loving microbes.
Oh, thank you. Sam already knows me and knows that I sometimes have only one working brain cell. So yeah, acetophiles. I kind of like that they do survive in like really acidic environments. If I think like pH 2 or lower, I mean, I think it's pretty cool. So I would go smack in the middle. Let's say a 3.
All right, acetophiles coming in at number 3. We're going to go next with radiophiles. So
oh my God.
The radiation surviving, loving microbes.
I think that is the first time that we're doing this kind of ranking where I hear something and I go, number 1.
Number 1.
No doubt about it.
No doubt about it.
And the coolest one is the fungi that are growing in the inside walls of Chernobyl. Yes.
See, I was thinking tardigrades.
Oh, yeah.
I had that. I wasn't even thinking fungi. It's fine, folks. I have no idea how to pronounce that word.
Nobody does.
The F word. That's just, I'll affectionately call it it's the F word.
They who should not be named.
Yes. If only I could find Voldemorto mycota or something.
Voldemort.
Voldemort. Oh, that's good. All right. So we've very quickly taken the 1 spot with radiophiles.
Yes.
Thanks, Chernobyl. Up next, and then acetophiles coming in the 3. Next category is going to be halophiles. So the salt-loving microbes.
Yes. Given that I'm a salty popcorn kind of girl, I can get behind the love for salt in general. So I would say let's fill up the spot number 2 right there.
Wow, we got 1, 2, and 3. You got our last 2 competing for last place. And so up next, we've got barophiles. So these would be microbes loving that high pressure.
Honestly, that's also pretty cool. I'm thinking like hydrothermal vent kind of guys that live like deep under the sea, which is pretty cool. I mean, we only have spot number 4 open because spot number 5 we're going to toss, so 4 it is.
Fair enough. And then coming in last place, the type of microbe that Julia hates the most, folks, is
Other than bacteria.
are the thermophiles.
Oh, there we go. Yeah,
Thermophile being number 5.
that's a good spot at number 5. I was scared that it would be like, because thermophiles are the ones that like warm temperatures.
Mm-hmm. Yeah.
I don't understand, neither in humans nor in animals, why you would do that. Like cold temperatures is where we are good at.
Yeah. Okay. Interesting. So we've got radiophiles at the 1, halophiles at the 2, acetophiles at 3, followed by the barophiles in 4, and then last but certainly least are the thermophiles.
I can
think
get behind that.
for me, I'm putting thermophiles at 5, mainly because I don't find them that special. Probably because, in my opinion, they're sort of easier to grow. Like we'll just toss a sample in. We got people that want something that, it grows at 55 or 60, and things seem to grow. I get that if we're talking about 80, 85, start getting up there, it's probably more cool. Obviously,
True.
Taq polymerase from earlier.
Oh, yeah.
Thank you for that, Yellowstone National Park. We love those bacteria. But in general, I think I'm putting thermophiles at 5, and I think I'm going to have to put halophiles at 4.
What?
Yeah, I think that's got to drop for me. Again, if I'm talking about purely just the complexity, it's like, you just toss in a bunch of salt in the media and it's good to go. I was really convinced at one point in time in my PhD that I'd isolated the most halophilic bacterium like ever.
Wow.
And I thought that for about 18 minutes. And after a quick Google search, I discovered that was definitely not the case. But it was growing in. It grew in up to, I believe it was 20. It was either 23 or 24% weight per volume sodium chloride. Mm-hmm. It was very, very salt loving in that regard, and so I got super excited.
Do you happen to remember by any chance what the most halophilic would survive in?
I don't off the top of my head, but I do recall the most salty body of water on planet Earth when I looked it up. And any guesses where that might be? The most
I would
salty
guess
body.
in some desert, maybe.
Yeah, you're not far off.
Or some mountain range in like deep below the mountain, maybe.
Yeah.
I don't know.
Well, it is in Antarctica.
Oh.
As far as I know,
true.
unless it's changed. I could be wrong, so we're going to go with what I remember from 3 years ago, which is not much. But I believe it was called Don Juan Pond down in Antarctica. And
That's a great
essentially
name.
there was a bunch of like calcium chloride in the rocks around this particular body of water. Mm-hmm. And so this pond, I believe, to the best of my knowledge, it was very shallow, maybe like a foot deep, just a few inches deep, and the rocks around it, the geology around it, really rich in this mineral, and it keeps leaching into the water, so much so that it remains liquid year round, and so it doesn't freeze. It doesn't freeze at all, and but it's so salty. And I recalled reading about metagenome analysis, whatever, of the Don Juan Pond water, that sort of thing. And I can't remember the exact percentage, but it was like 30 something. It was a whole mess load of calcium chloride.
Wow.
And but yeah, it was So for about 18 minutes, I thought I was in line for the Nobel, and then Google sunk my battleship. So I'm putting the halophiles at 4, and then I think I'm going to put acetophiles at 3, like you. I love me a good acetophilic bacterium, and I enjoy the cultivation process with them, mainly because I like that the media changes color. Like as I'm messing with the pH, it gets really light, or you crank it up alkaline, it gets really dark. But yeah, acetophiles, and kind of makes the auger. It's a tricky process when you start getting down below, say like pH 5, the auger gets really, really soft and mushy.
Ooh.
And so you've kind of got to do some things with it to
What kind of things?
I just knew you were going to ask that. And I'm not really sure I have the answer in front of me.
Okay, sorry, it was so suggestive. I thought, what kind of things are you doing to your auger?
Yeah, I think it has something to do with adding more salt and then adding also in a higher concentration of agar.
Oh, to help
Yeah.
with the structure probably,
Yeah.
because it would fall apart, right?
Mhm.
Yeah.
Yeah, but it's almost like Jello. It's very, very easy to rip at a pH of 5. Not that I've done that recently.
You're ripping your agars.
Oh yeah,
Okay.
at a pH of 5, 100%. I'm an
When
auger
you
excavator.
strike, like when you.
Yeah.
Oh, okay.
Yeah,
I was like, what are you
just-
like? You take it in your hands and you rip it apart like a piece of paper, and like, what are you doing in your lab?
No, it's just so mushy, and especially if you're using like a metal loop, it just right in and just scrapes all the way through it. I've yet to streak out like a very acidic medium like that winds up pretty at the end of the day. And then I think I'm going to put barophiles at 2, because I think the idea of something living, in the trenches at the bottom of the ocean, that's sort of where my brain goes. I'm like, that's super cool.
That is cool.
Yeah. Yeah. So if you're a person out there that likes to scuba dive and you want to collect some sediment off the mid-Atlantic ridge or something like that, hit us up. I'd love to have that. Don't get eaten by a shark. And then, yeah, in the one spot, I got to agree with you. I'm going radiophiles. I think the idea that you can survive, intense amounts of radiation is, I mean, that's pretty spectacular from a life standpoint. It's not something that I'm going to handle very well. But, yeah,
And it also makes so much sense that there has to be organisms that can do that, right? Because radiation is like a common hazard of this world, less so, but everybody, like everywhere else. That is so cool.
Yeah.
Makes you think, is there life on other planets?
Ooh, there we go. We'll get into that question another day. But
One can dream.
One can dream.
One can dream.
Yeah, that's true. Oh, my. Well, Julia, I appreciate you playing along and handling those blind rankings so well. I felt like you did so with an air of elegance and grace, dare I say.
Well, thank you for preparing those. You did a really good job. Like, I thought those were pretty fun.
Yeah, well, I attempted to have them not be horrible.
Succeeded.
Yes. No, totally so. So that was good. I think I mostly agree with you. Yeah.
Yeah, that's good. Nice.
Well, Julia, I think we have suffocated these people perhaps long enough here today. And we appreciate you holding your breath with us, folks, as you survive and you trudge through this anoxic mire that is the brains of Julia and I. We do have, I suppose, a teaser here in the works, and that is that we have officially secured our first our first interviewees. And so I think in the next few weeks we will force them to sit down with us and actually get some interviews going on here on Life Unculturable.
Yeah, I'm excited to introduce some more people to this chamber and our audience.
Yes. Oh, my thoughts exactly. Couldn't be said any better than that. I think it'll be strange having other people in here with us, but I'm looking forward to it. I'm like, where are they going to sit? Are they going to sit? Stand, peasant. No.
It would be hard keeping the professionalism that we clearly do not have.
Yeah, I'll have to wear a bow tie that day. I'll have to step up my game.
I mean, you're kind of nicely dressed today too, with like a nice shirt.
Yeah, well, this is meeting Sam. I've got a meeting this afternoon, so yeah, I know.
Sam.
I got my meeting Sam get up on. So
oh, nice.
Yeah.
So we want to get meeting Sam meeting ready.
Yeah, that's probably so. That's a good frame of mind to be in. Is meeting Sam needs to be meeting ready.
Well, by all means, I think it's time to go back to the lab.
All right, back to the lab. I couldn't have said it any better, Julia. It's been a blast, and thanks for hanging out with us here on Life Unculturable. Oh wait, are we going to say it together? Life Unculturable. Should we do that? Did we do that last time?
I don't know. I can't remember. I'm not there yet with the recording.
I think maybe. Maybe. You want to try it just to have one?
Okay.
Okay.
1,
okay. 2, 3.
Life
Unculturable.
Oh, we're getting better at this
Boom.
every time.
I know. Look at this. We got this thing going within like 5 minutes. We're succinct.
Nice.
They'll come in next week and it'll be like an asteroid hit the dinosaurs. We'll be some kind of little rat running around trying to get bigger.